Hi, everybody. This is the International Academy for Clinical Hematology Journal Club. It's 6 p.m. in Paris, 5 p.m. in London, noon in New York. And it is my great pleasure to welcome you all to this new broadcast as part of the Journal Club series brought to you by the ICH. And actually, I just realized that our first journal club, the first ICH journal club, was in October 2020. So we're celebrating this month our fourth anniversary. And thank you all for being loyal and for following the ICH activities. So today's journal club is a relatively special one, I would say. As you probably know, we usually discuss original papers, original studies. However, the Scientific Steering Committee has chosen this paper dedicated to targets and treatment in primary CNS lymphoma. Actually, we've been discussing within the committee for several weeks that we needed to to discuss the CNS lymphoma treatment, but we didn't see a recent paper. This is why this review was a very good opportunity. It's a very nice, comprehensive review, which actually discussed the current treatment algorithm in primary CNS lymphoma. But also, I think most importantly, it has handled the future perspective, future venues, signaling pathways that one can investigate in primary CNS lymphoma, but also in relapse refractory lymphoma. And you have a very nice table in this review summarizing some of the most promising agents being investigated in clinical trials and also explaining their mechanism of action. You have actually a very beautiful, colorful figure actually looking into the different signaling pathway, because the NF-kappa B has a key role actually in the proliferation and the development of primary CNS lymphoma. You would appreciate that targeting the BCR pathway, the toll-like receptor pathway, or the PI3 kinase AKT mTOR pathway are all of them very promising. And obviously, we're looking forward to see data looking into BTK inhibitors. Of course, the mTOR inhibitors, the PI3 kinase targeting agent, but also we have some strong evidence about the involvement of the IRAK4 pathway, and there is a drug, namely imavusertib, being developed, being tested not only in primary CNS lymphoma, but actually in also in acute myeloid leukemia. And of course, I don't want to ignore the famous IMiDs, especially lenalidomide, pomalidomide, but maybe at some point the new cell modes, iberdomide, mezigdomide, which act on the IRF4 signaling pathway. And to discuss all this, we're very fortunate tonight having with us Professor Carole Soussain from the Curie Institute in Paris. And actually, Professor Soussain, who's today suffering from a nasty flu. So I'm very grateful for her dedicating some time despite the flu. Professor Soussain has really dedicated all her career since her fellowship to CNS lymphoma. And actually, if you look into the literature in this field, you will immediately realize the seminal developments and contributions she has made. We owe her for instance, the role of auto in CNS lymphoma, the development of some of these conditioning regimens. And she has been involved in almost all drugs and trials I'm aware of in this field. So, Carole, thank you for joining us. Welcome to the ICH Journal Club, and congratulations for co-authoring this review. And of course, guys, please don't hesitate to jump in if you have questions to Professor Soussain or comments or suggestions. I'll do my best to share them. So my first question to you before going into the details of what's in the review, maybe if you can give us, Carole, a summary about the current treatment algorithm for CNS lymphoma, both in the front line and in the relapse refractory setting. I know it's a very broad, large question, but maybe the key concepts, the key steps.
Yes, first of all, thank you. Hello, everybody, and thank you, Mohamad, for the introduction. Yes, of course, I can give that, although it is still a changing field. But there is a consensus, even a European and international consensus on that. So first of all, there is no doubt about using high-dose methotrexate in first-line treatment for induction. And the use of rituximab has been a little bit challenged by a phase 3 trial, but in the real life, most of the centers still use rituximab if you listen to the experience of colleagues. Sometimes with different modalities of using rituximab, like more often or with higher doses. And then what has really changed the last years is the use of consolidation with intensive treatment in first-line treatment. First, it has been developed in relapse, but now it is really a consensus that consolidation in complete response patient is better with intensive treatment. And the recent data is that we can extend the year, so we don't limit the intensive treatment at 60, not even 65. We can even do to 70 and even to older patients, like 80 in very selected patients, of course. But there is a paper recently published paper from the German group, and they published the feasibility of intensive treatment in patients over 70. So, but of course it is in selected patients, but when we can, we tend to offer these chance of treatment. So this is very, so then regarding the induction treatment, there is no consensus about what combination. So what I always say that we don't need to fight about what combination, because they all give the same kind of bad results, because the rate of complete response is not above 50%. So they are all about the same, and they are not optimal. So there is no consensus. And if you read there is the ESMO guidelines that have been published. This is just high-dose methotrexate combined with other drugs, the one each center is used to, because we're used to the side effects and how to use it. So that's the main consensus.
So, Carole, what are the results today in terms of progression-free survival and outcome for those patients receiving auto transplant? What is the PFS rate and overall survival rate?
For transplant in first line?
Yes.
So when we looked at in the results in our database, we have a national database with thousands of patients, and we looked at it a couple of years ago. I don't exactly remember when, but we had about 600 patients who received a transplant, and about a third that received transplant at first line. So we're looking at the survival of patients who had received not in intention to treat, but who had received intensive treatment. And the overall survival was about 80%, with a longer survival, 70, 80%. And we really see the best prognosis when transplant is done in first line. And then it's still good in second line at first relapse, with about a long-term survival of 50, 60%. But then if you wait for the second relapse, the overall survival will drop. So yes, we have very nice survival curves in first-line treatment, but the problem is to increase the number of patients who can proceed to intensive treatment. So we really need to work on the quality of the induction treatment and the criteria for response, which is another.
So that's actually, that brings me, Carole, to one of the questions from the audience about the criteria for eligibility to intensive treatment. Because this disease is very tricky, because when they arrive at diagnosis, the patient can be in a very bad shape. But actually, when you start treating them, they can improve. So initially, they are not eligible, they become eligible. So how do you do it in real life? What is the kinetics of the process?
Well, we see how they improve with the treatment, and they won't be eligible for transplant. It's rare. Under 65, it's very rare not to be eligible for transplant. And we look at comorbidities. If they have, we don't have strong and robust criteria. We don't use that, but we look at comorbidities, kidney, liver, and heart, cardiac function. And usually they improve. Then we can discuss the indication of transplant if they have very severe cognitive decline, if they don't understand what we are doing for them, if they didn't improve on that side, or if they have, if they are at risk of complication from the resting situation. But it's quite rare. It's quite rare, actually.
So another question, when you proceed to auto-transplant, what is the recommended conditioning regimen or high-dose chemotherapy combination today?
Yeah, so for that also there is an international consensus, and it has to be based on high-dose thiotepa. Then there are different kind of thiotepa-based intensive treatment combined with busulfan and cyclophosphamide, but we use at the beginning in relapse, or without cyclophosphamide, or with carmustine. And two retrospective studies, one from the EBMTR and one from our database, really showed, it's again retrospective, really showed that the beam conditioning regimen results in lower survival, in lower results in retrospective studies. But in our study, for example, it was not, the bias was not, there was not really bias, because it was just center dependent. Some centers decided to do beam because they wanted, and the other centers did the thiotepa base, and the patients treated with beam did a lot worse. So it's thiotepa-based.
So when you say thiotepa base, is it about the team regimen, for instance, sort of an equivalent to beam, or is it thiotepa plus busulfan and VP16?
Yeah, it's more, we have more experience with thiotepa and busulfan, or thiotepa with carmustine. We usually keep thiotepa carmustine for elderly patients, and we can adjust the dose of carmustine of thiotepa and busulfan also in elderly patients. And in this paper I referred to earlier about the German experience, they use a thiotepa busulfan in elderly patients with lower dose of thiotepa and lower dose of busulfan. And they were using 5 milligram per kilogram, 2 days of thiotepa, and busulfan 6.4 instead of 8 milligram per kilo that we use in elderly patients.
Well, we have plenty of questions actually about this transplant issue, because I guess this is motivated by the very high survival rate. So what is today the transplant-related mortality in this population? Because I guess it's slightly higher than what we usually see in other populations.
Yeah, clearly when we use the TBC, the thiotepa, busulfan, and cyclophosphamide, kind of the first regimen we use in relapse with very good results, we had about 7% mortality in relapse refractory patients. And this was found in our experience, but also in the US experience. They've been using TBC a lot. But this was in relapse patients. And also another observation we made in our retrospective study, that the non-relapse mortality was higher in pre-treated patients in second or third line when we use intensive in second or third line treatment. Otherwise in our previous study with prospective study with the PRECISE trial, we had a high rate of mortality, like 10%. It was a multicentric study. It was unusual to have this 10%. But usually it's about 7%, and now we drop the cyclophosphamide in our first line treatment. I don't know the results, but it really seemed a lot less toxic without the cyclophosphamide. So we might be, I don't know, I don't have the results, but we'll be lower than that.
Excellent. Thank you very much.
With the thiotip-acarmustine, it's about 4%, or like the BEAM regimen.
So what about the patients who are not candidate to intensive chemo? What are the current treatment options? First line.
So first, we really insist to be sure they are not candidates.
Okay.
Because they are center dependent. So really insist. And then we don't have a lot of options actually, because so far maintenance treatments are we don't have the results. There are some studies ongoing or that have just finished their recruitment, but we don't have the results. So we don't know yet if the maintenance treatment has a benefit. That would be for elderly patients, the maintenance treatment. We expect to have less relapse. Otherwise, the PFS is about, the median PFS is about 1 year in elderly patients with no consolidation. So there are several ongoing studies with maintenance, either with methotrexate-based regimen or with ibrutinib. There was an Israeli study with ibrutinib. They show encouraging results in the first preliminary report in elderly patients, and with lenalidomide and ocarbazine also. But we don't have the results yet.
So one question we have from the audience is about the role of radiation. Is this something that remains common practice? How should it proposed and when?
So in France, I will give our guidelines. We are very radical for irradiation, against irradiation, I would say.
Okay.
We don't use anymore the 40 gray consolidation. We don't use that because we really showed, several studies show that it is very toxic even in young patients, so we don't use that. We can sometimes use in young patients, incomplete remission, not eligible for transplant, we can use low-dose irradiation, 23.4 grays, that has been validated in a US study. In elderly patients over 60, the toxicity, even of the low dose, has not been completely proved. The non-toxicity has not completely been proved. So in France, we just don't irradiate over 60. And we don't offer irradiation as a treatment for relapse, because then you have to use high dose and it's toxic, it can be very toxic. And because we have other options. But I know that in other countries, and you find that in the European guidelines, they can offer irradiation in relapse in patients who are not eligible for chemotherapy.
Yeah, actually, I was referring because the key contributors to this field are also our German colleagues and the Freiburg group, and obviously they include brain irradiation in many of the protocols, but your reluctance is mainly related to the toxicity over the short term, but also the long term. Did I get it right?
Yeah, short term and long term, of course, it can be just a nightmare to have patient in complete remission, but completely like with degenerative disease. So we really don't use that. And teams that who use the intensive treatment, they don't use irradiation as part of the treatment. We don't do irradiation after transplant.
So before moving to the relapse setting, I'm taking one question which I think is interesting. So beside chemo and the issue of transplant, what is the role of rehabilitation in these patients, and what's the importance of the non-medical, I would say, non-drug based approach?
Thank you for this question. It's huge. It's huge. And the baseline evaluation, the neurocognitive evaluation is part of the baseline workout. So we have to do that when we can, before. And then there is the cognitive rehabilitation and also the physiotherapy. It's major. Yeah, we need that because if you leave a patient with a motor deficit, not doing physiotherapy, it really worsen, and then you can sometimes not do the transplant just because it's not in condition. So it is very, very important. And we usually do the cognitive assessment. In our recommendation, we recommend to repeat the neurocognitive evaluation like once a year when we can, to detect the long-term toxicity.
Excellent. Let's move to the relapse refractory patient. So let's take a scenario, and you may tell me anyway, once they have relapsed, they are all in the same basket, but someone relapsing 2 years or 3 years after auto transplant, and another patient relapsing 1 year, let's say, without auto. Are there any standardized treatment approaches, or it's just empirical based on the local experience? What are the most popular options, let's say?
Well, there is no standardized salvage treatment. If patients are fit for eligible for chemotherapy, we usually offer second-line chemotherapy, just like DLBCL, systemic DLBCL. And we can sometimes repeat the methotrexate if they relapse a long term after the first-line treatment, and if they were in complete remission after high-dose methotrexate. So we can either repeat the methotrexate or use another chemotherapy, just like high-dose RSC-based or high-dose ifosfamide-based chemotherapy, like RDHAP or ICE. And then the question will be, is the patient eligible for transplant or not? So it was easier, maybe this question earlier, because less patients received high-dose chemotherapy in first-line treatment. So if they are still eligible for transplant, we recommend a transplant if they're eligible. But if they have been transplanted first-line, we have two options. Either the relapse occurs early after the first line, but it's very, I would say, French, because we can do that. All the countries cannot do that, but we can discuss the CAR T cells, because we will be in the indication of early relapse or of a DLBCL. And we had some encouraging data. So if in case of an early relapse after transplant, we will consider CAR T cells. If it's a late relapse, or if the patient is not eligible for CAR T cells, and if in case the patient had received intensive treatment, we will offer chemotherapy and discuss low-dose irradiation as a consolidation. If the patient is not eligible for chemotherapy, then we have all the chemo-free treatment. It's completely off-label, but we can use ibrutinib, lenalidomide, temozolomide, mainly those drugs.
So we have one question about at time of relapse, if you have a patient who received the first auto and who's still fit, is there a room for a second auto?
Not after high-dose thiotypa.
No.
It has been done. There are a couple of cases. I know that from a German colleague, that they did the TBC after thiotypa carmustine, or after we've done that maybe a couple of times, but less than 5, I would say, after BIM. But usually we don't do. But after BIM, we have discussed that a couple of times.
Okay.
But after thiotypa busulfan, it's hard, and you're happy not to leukemia the first time, so you don't want that with the second time.
Well, I guess toxicity is going to be extremely high, and we already know it's relatively high compared to other indications. So before moving to the most promising agents under development, my question to you is, I mean, you guys involved in this very particular disease space have done a great job over the last 20 years with dramatic improvement. But from your experience, what are the barriers to develop novel treatments in lymphoma? Because in CNS lymphoma, because when I look to the other lymphomas, actually, there is a plethora of trials and everything is being investigated. And this is good news because things are improving quickly. So what are the barriers which are intrinsically specific to CNS lymphoma?
Well, just think about the incidence of the disease. In France, we have about 300 new cases per year.
So
So
ultra rare disease.
it's a rare disease. I think it's the first barrier. It's hard to do. There are two main. First is the non that the industrial are not interested. They were not interested in this niche because it's not a frequent disease. They are becoming a little bit more interested now, but for years they were not interested in developing new drugs in this disease. So it was really a struggle to get a new molecule for this disease. It is a little bit different now, but it's still rare. So we cannot conduct several open phase 2 studies at the same time. It's hard to do phase 3 trials unless we will do European or international studies. So that's the first. Then I think there is a real lack of knowledge about the biology because you know that the biopsy are very small. They are collected by stereotaxic biopsy, so it's nothing to do with the lymph node. So we had a lot of, we are very late. We were late in the knowledge of biology. It's changing. There are some very good teams working on the biology, but it's still exploratory. So it's not routine to do the molecular biology. And the last studies published, I think last year, the year before, they showed that actually the CNS lymphoma is a very heterogeneous group of disease. It's not only a DLBCL non-GC type. There are different subgroup of patients. So we will need to probably know better about that and adjust the treatment, especially for the target treatment, to this subgroup of patients. But we need to, get the results of the biology earlier or surrogate markers so we can adjust the target treatment. Then it's difficult to see. Also, we have other challenges that treatment. We don't know a lot about prognostic factors. So this is a huge barrier. We need to work on the prognostic factors of biomarker for surrogate marker of response. Probably the imaging. The MRI is not optimal. If you look at 2 MRI, they won't show any gadolinium intake. One patient will relapse, the other one will never relapse. You don't understand why. So there are a lot of things to do. I think these are barriers.
No, I think these are really serious barriers, but despite this, we would like to continue to progress there. So my next question is, and this is very nicely summarized in the review, and there's a table about it. What are, in your opinion, today the most promising agents under development? This is where you would like to invest energy, resources.
Well, as I just comment a little bit, the target drugs are probably good, but we need to know when and how to use it. Because if we use IBTK in a patient who doesn't have this signaling pathway, this activated, we doesn't need. So first we have to better know what patient will benefit from target therapy. And then I would say CAR T cells. I think that all the work we've done with intensive treatment will have to be repeated with CAR T cells.
But the problem with CAR T cells is that access is very limited. in the majority of the countries and the colleagues following us at the ICH, especially from low middle income countries, but even in rich countries, only a few centers. So we'll talk about immunotherapy, but I would like to maybe highlight among the current available treatment options, as you said, and I do appreciate we need to have a sort of a biological rationale for
Yes.
this. And you mentioned the limitation of the biopsy. So it's very difficult to understand for a specific patient, given the heterogeneity you alluded to. Not all patients are the same. So beside CAR T cell, and we'll talk about CAR T cells, and actually we have a question about this. We have even a question about allotransplant from Dr. Vaitikanite. I hope I'm pronouncing well. What are the experimental agents currently under development?
Well, we are testing the addition of ibrutinib or lenalidomide to the high-dose methotrexate in induction because, as I said earlier, we really have to work on the induction treatment to increase the rate of patients who can go to intensive treatment. So that these two class of medication, IBTK and IMiDs, were the most studied first in relapse. So now there are studies with this drug in first line, as it is shown on the table in the paper. So I would say these drugs, in addition to high-dose methotrexate.
So what
So,
about the IRAK4 signaling
well,
pathway? Because this for me was a surprise because before reading this paper, this review, I was aware about targeting IRAK4 or IRAK4, whatever. I don't know how to pronounce it. In French, we would say IRAK4. In acute myeloid leukemia with some very promising ongoing phase 1, phase 2 trials. So I was quite surprised to see this in CNS lymphoma. So what do you think about it?
yeah, but it's a way of inactivate NF-κB by two different pathways. It's like a little bit using maybe IMiDs and IBTK. So I think I was surprised to I learned about the IRAK4 because I didn't know about this molecule before this study. We opened the study in our center. I didn't know it was involved in the toll-like receptor signaling pathway, but I don't have results yet. So, but it is interesting because they use it with ibrutinib, and they say that the company said that it can even reverse the resistance to ibrutinib. I don't exactly understood how it can reverse, but at least it can inactivate the NF-κB by two different ways when you use IBTK plus the IRAK4 inhibitors. And according to their results, it could also reverse the resistance to ibrutinib. So we need, it's a newer molecule and it's under study. I have absolutely no results so far. They have very few results on CNS lymphoma. But for sure, as an on single agent in relapse, we will have maybe a proof of concept, but I'm probably sure that it won't cure the patients. But at least if we have a proof of concept that there is an effect in the CNS, it would be already a good effect. And it's a small molecule, so it can probably reach, that was one part of the advantage of this molecule, that it can reach the CNS.
Excellent.
So let's see.
No, very good, very promising, and it's always good
Yeah.
news to have new things under development. We have an interesting question here because we're talking about the activation of the NF-κB signaling pathway. And one question we have, and this is not mentioned in your review, is about the proteasome inhibitors, because we know, for instance, that proteasome inhibitors are used in myeloma where the NF-κB signaling pathway is very important. Are there any trials, combinations, or this is something that hasn't been tested yet?
No, and that's exactly an example of the barrier. I don't know if you, many years ago, it's an anecdote, I'll tell you. Many years ago, I saw an abstract, you might know that better than me, that in myeloma with CNS involvement, they had encouraging results in patients who had received a proteasome inhibitor.
Absolutely.
Yeah, so I saw that. I immediately contacted the firm, met them in Lugano, talked to them about how interesting it would be to develop this drug, to try this drug, and never heard back from them. So I'm not aware of any studies with proteasome inhibitors. And I
Okay.
think that there is no.
Excellent. So a few minutes ago, and we have a question here, which I think is an important one. You mentioned the promise of CAR T cells in lymphoma, and you mentioned very nice results in the relapse setting. But the question we have is, why don't we move them into earlier line, into the frontline setting, especially that the tolerance is likely better than the high-dose chemotherapy and auto-transplant. Is this something you are considering, or maybe you have already tested it in a few patients?
Tested, no, we haven't tested it, but we are considering it for future trials. And it's exactly the same discussion that for transplant. We started to use it in relapse, and then we moved it to first line. So and I think we'll see that in the next 5 or 10 years, we'll probably select, for example, the patients who are primary refractory, and there are like about 25% of patients primary refractory to methotrexate. These patients have a very bad prognosis. So we can imagine to use CAR T cells earlier for these patients instead of waiting till the transplant. So yes, but that's the work for the next 15 years, I guess, or
Well,
even for patients, elderly patients not eligible for transplant. We could eventually consider CAR T cell for this patient in first line. But yes, of course, it will be a challenge.
superb. No, this is very clear. And actually, I think you're providing us with some very optimistic
Yes.
inspiration. So my last question, because we're reaching almost the end of this journal club, we try to stick to 40, 45 minutes maximum. If today you have all kind of drugs available, access to radiation, and no money constraints, Carole, what would be the ideal clinical trial you would love to see in these patients? What is the treatment? Something where you believe you are going to likely modify the natural history of the disease of the patient?
That's a difficult question, but that would be collaborative study, probably, at least European or international study. And I will think about probably like a basket study, different arms, because we have several questions. And so I think we should go for a study that will, with the bio. So I can imagine that we will have the biological results, of course, in the week. It's in a
Absolutely. You have no, I mean,
we're
all
just breathing.
resources are available. Absolutely.
We have 5 minutes of dreams. Okay, so let's imagine we have the molecular biology results in about 10 days. So we can test the addition of a target therapy according to the results. And then I think that we'll have to work like in systemic DLBCL, driven response strategy, response-driven strategy, like, because I'm not sure we need to transplant all the patients. So we need to work on the criteria for response, the prognostic marker to exactly identify patients who have to go to transplant or the patient who can avoid this intensive treatment. That would be biology, target therapy, criteria for response, treatment adjusted to a prognostic marker that we don't have so far. We just work with the eligibility, general status, age, but we don't have the IPI, we don't have all the prognostic score. So score, response adapted driven strategy, and biology. It's a lot of dreams.
Well, of course, everything starts with a good dream, but
Yeah.
also with passionate people like yourself, Professor Soussain. It has been a very inspiring and inspired discussion. Thank you so much, and thank you
Thank
all
you.
for really asking great questions. I do apologize. I wasn't able to take all the questions because it's not easy. We need to structure like a sort of a natural flow for this journal club because this is being broadcasted live, as you know. So I hope you have enjoyed it. I hope you got inspired to put more resources, more energy, more time in these rare diseases because obviously the patient deserves the best wherever they are, and having a rare disease is, we know, is something crucial where we need to invest more resources, more energy, more collaboration, and this is exactly the goal of these educational activities brought to you by the ICH. So I hope we have provided you with some valuable insights, and we maybe help to foster stronger collaborations. But I'm very pleased to hear that despite the complexity of CNS lymphoma, there are very promising new agents, new candidate being tested, different pathway, the BTK inhibitors, the IMiDs, the immune therapy, cellular therapy with CAR T cells, targeting the IRAK4 pathway and the mTOR pathway and many others. So thank you so much, and I
Thank you.
hope to catch up with you guys very soon. I think we do have another activities this week on a Thursday. Until then, please stay safe and keep well. Take care.
Thank you. Bye.